friends, today I'm going to give you the different method of RNA isolation and purification technique. Without delay, I will going to let you know from the first step. First technique, that is guanidine acid phenol extraction technique, which is also known as TRIzol or Reagent method of RNA extraction. In this technique, guanidinium salts act as silica and inactivate RNAs, and phenol chloroform act as a separator RNA from the DNA and mixture of other protein. To perform this method of RNA extraction, first step is to lyse cell. So to lyse cell, we use TRIzol to lyse cell and add chloroform afterwards, and we will mix with pipette, and then there will be a development of 3 layers after centrifugation. At the top there will be RNA, in the middle there will be protein, and at the bottom of tube there will be other proteins and impurities. So we collect the RNA from the top layer and perform precipitation of RNA with the isopropanyl alcohol, which is 100%, and washed with ethanol and dissolve RNA. So RNA will be in pure form. The pros of this TRIzol method of isolation of pure RNA includes RNA will be high yield, and this works for the most sample types. But the demerits of this technique of isolation of RNA is there is a use of toxic chemicals like TRIzol, chloroform. So we have to be aware on that, and it is also time consuming process. We have to perform repeated centrifugation, mixing with chemicals. Now we have another technique of RNA isolation. It is Silica technology, and it is also known as a glass foil filter method, and in another terms RNeasy and variants method. The principle of this silica technology is that RNA bind to the silica membrane in a high salt condition, and the procedure of this techniques includes sample will lies with chaotropic salts, and that will load into the silica column. Afterwards, RNA binds and contaminant will flow through the column, and in the wash steps impurity will be removed and RNA elute in pure form with water. The process has benefits, like this method is quite faster as compared to TRIzol method, and safe and reproducible as well, because only there is a addition of silica membranes to separate RNA from the DNA and other impurities, and this is the most expensive method as well. Now a third method of RNA isolation is density gradient centrifugation process, with the addition of cesium chloride which plays a vital role for the extraction of RNA. And RNA is separate based on its density during ultracentrifugation. So the process includes initially neutralize the cell, and sample will be loaded in a cesium chloride gradient, and there will be a ultracentrifuge for several hours, and RNA will collect in the form of band. The approach behind this process is it is extremely pure RNA and has a minimal DNA contamination. But the demerit of this techniques of RNA includes it requires ultracentrifuge, which could be costly and very time consuming to centrifuge. Beside that, we have another method of RNA extraction as well. This is the fourth method, magnetic bead technology. The principle behind this magnetic bead technology is RNA bind to magnetic beads and coat with the silica or Oligo DT. Magnetic bead technology includes the lyse of sample initially, and we add magnetic beads, and RNA bind to bead, and magnet pull the bead aside, and we wash and elute RNA. So pros of this technique includes automation friendly and high producibility, and the cons of this method is it is expensive reagent. Now the fifth method of RNA extraction includes the lithium chloride and urea isolation method, on which the sample lithium chloride will selectively separate RNA from the sample, and DNA and protein remains soluble in the solution. So procedure includes of initial extract of nucleic acid, and there will be addition of lithium chloride and incubate at a low temperature. That will gives another steps to separate the RNA with the centrifugation in the platform. Then the pallet of RNA will be washed and dissolved. RNA will be in pure state. So the pros of this process includes it is very selective for RNA and remove polysaccharide as well, but the cons of this process is it is not suitable for the small RNAs. Now the ultimate and last techniques for the RNA isolation is Oligo DT cellulose column chromatography. Principle of this technique is Oligo DT binds poly tail of a messenger RNA. So the process includes isolation. Initially RNA pass through the Oligo DT column, and messenger RNA binds with the poly tail, and that ribosome RNA and transfer RNA will wash out away and elute messenger RNA in transcript form, and the approach will be high specific for messenger RNA, but the cons include does not isolate total RNA, and this process is also costly. Thank you so much for this time. I will come with another methods and the techniques for the DNA isolation in the next video. Thanks for watching.